Thank you very much, Priyadarshini, and thank you, Prashanta. And you can hear me and see me, right? My slides, etc.
Yes, we can see you.
Yeah, wonderful. And it's lovely to speak with Maheshi and Nilika both in one session. I think Maheshi is in Oxford at the moment, and both Nilika and I had connections with Oxford because we were students there, and it's so lovely to take part in a sort of a session like this. So what I'll do is extend on the topics or the areas that Maheshi spoke on and just speak about mixing vaccines and booster doses. So we all know the virus that we are dealing with, sort of spike protein directed vaccines at the moment, and about 8 vaccines have been approved. Some have been sort of rejected, abandoned, and the take-home message is not every vaccine that was developed was a success, but there were some vaccines that had to be abandoned during the process. Vaccine rollout has occurred, and Maheshi clearly illustrated that many countries have vaccinated big numbers of the population. Sri Lanka has done the same, but there are countries that are lagging behind. You can see Bangladesh, Nigeria, etc. And until we vaccinate all these population to an appropriate level, you're going to have issues because variants will arise and then put the vaccine program across the world into a bit of an issue. So this has to be considered and has to be taken into consideration. That is why I think the WHO is pushing so much to have some sort of consideration in that aspect with regards to vaccine rollout. So I will touch on just a quick overview of the immune response and then go on to mixing vaccines, immune deficiency, the group that I deal with, and booster doses of vaccines. So, you know, there's a whole range of immune response changes that occur with COVID. Certain cells increase, cells decrease, cytokines vary, hypercytokainemia, etc. And the strategies have been designed to try and deal with these and mitigate these and neutralize these so as to get the patient not get into ICU and not dying. You have the immune response coming. It takes some time. That is why you can't get everyone getting COVID and trying to get the immune response because, and why we use vaccination with social distancing public health measure. You have the T cells, the CD4 and CD8, and you have the immunoglobulins across the range. Certain immunoglobulins don't come into good levels in certain areas like the lung, and that has to be considered because when we treat patients with immune deficiency, we take that into account. So this is the immune response that occurred. We have to also remember that the oral antiviral agents and the monoclonal antibodies are coming into focus, especially in patients with immune deficiency, because sometimes the immune response are not sufficient. We do need additional measures. So those 4 measures, public health measures, vaccination, together with the monoclonal antibody, which can be a bit expensive, but given in the correct group of patients, it's very effective, especially in our immune deficient patient, together with the use of the new use of the antiviral agents, the molnupiravir, etc., that is coming into focus, would be a good sort of a combination to try and deal with this pandemic. So next we go on to the process of the COVID vaccines. We know that range of vaccines, Mahesh spoke on that. There the mRNA vaccine, the viral vector vaccines, the inactivated vaccine. The different countries have used different combination of vaccines across their first and second doses. And we know that the effectiveness, efficacy, effectiveness varies, the cost, the storage conditions, etc., vary across those vaccines. Mahesh spoke about the efficacy in clinical trials, effective in the real world, but the vaccines have done what it set out to do, that's preventing severe disease, hospitalization, ICU care, and death. It has done that, reduced infection to a lower level, and both those infected people and those with disease can transmit the disease. So that we must remember, but vaccines have done what they have done as a first go, and now the process has to be consolidated. It changed the sort of pattern in the UK. This was early data, just around from December onwards when vaccination and so lockdowns came into the picture, and it was effective, but then the picture changed after that. Now, we have to remember that at one time, due to a very, very rare side effect with the AstraZeneca vaccine, certain countries in Europe moved out of the area and started using the mRNA vaccines as a second dose. So the UK persisted with AstraZeneca, but certain other countries moved and they changed their age groups, etc. First, they're giving it to the old over 60 years, and then they went to the younger people. So there was some maneuvering and movement there earlier on. And mixing vaccines is there can be homologous group or in the heterologous. That is, the homologous is either AstraZeneca, one of the mRNA vaccines, and the mRNA vaccine, I'll take Pfizer, and then the other one is AstraZeneca, Pfizer. So that was a question, a very, very topical question at one stage. Again, it came with Sri Lanka because, and many parts of the world, when there's a delay in getting back, there were not sufficient vaccines, and people wondering whether they could sort of mix the vaccines, etc. Should they wait? And there was a lot of discussion early on, and actually we have all spoken about this aspect. So the combined, so COMCOV study, which was published for the first studies, report came from the Oxford group, published in the Lancet. They looked at it. This was an interim study showing that there was reactogenicity, and these are the figures that were in the paper that was published in the Lancet. And there was a summary, it's more likely to develop mild or moderate symptoms as sort of reactogenicity. However, they were effective, they were safe, generally safe vaccines, effectiveness later. And people used to get chills, fatigue, feverishness, joint pains, malaise, etc. It was short-lived, no other safety concerns were there, and the younger group was getting the symptoms more prominently, and it was more prevalent in the young group. There was no hospitalization. Most of the reactogenicity observed 48 hours after the vaccination. Paracetamol use was higher. No problems with the hematology parameters, etc. And health worker absence for just 1 or 2 days was noted. So this was what was found in the preliminary sort of interim data that was published in the Lancet around, I think it was May. And then the next more formal data came. Maish was involved in the study where there was no safety concerns raised. There was some adverse event, but results support flexible use of a heterologous boost, prime boost schedule of the AstraZeneca and the Pfizer vaccine, and cellular and humoral immune response were comparable. So that was an important advance with regards to mixing vaccines. And at the same time, the Spanish group came because now the European groups were, European countries were using AstraZeneca first. Certain population had used AstraZeneca first, and they were getting their Pfizer vaccine as a second, or Moderna. So there was data coming out. This was the Spanish data, which showed that the Pfizer vaccine given as second dose in individuals produced an robust immune response and acceptable safety profile. So these were the immunogenicity and safety data, and I will talk about the effectiveness data, which has also started to come up recently. Then we had the two German studies again, which came up, heterologous vector mRNA boost vaccine induces immune responses and acceptable reactogenicity. The heterologous boosting was well tolerated, and it was comparable to the homologous mRNA boosting vaccine. And you can find the CD4 cells, CD8 cells were either comparable or higher in the mixed regime. This was the second German study, again showed real-world evidence supporting the safety and images of the combined mixed vaccines, which again came from another German group. You had in this study coming out, I think, was Sweden, which showed the combination of the AstraZeneca and the Moderna vaccine, again showing neutralization was high with the mixed regime. And this, again, small study, but it was against the beta variant, which was a troublesome variant, again, with regards to AstraZeneca, which they found in South Africa at one stage. So those were very, very reassuring data coming from the immunogenicity and safety perspective. And these were the more effectiveness data that has also come, a few papers. I've just selected a few papers from here. It shows that the heterogeneous heterologous AstraZeneca mRNA vaccines is effective alternative to increase population immunity. This was from Sweden that came up, and then you had the other studies coming from both from France and Denmark, which again showed a reduction in risk of severe SARS-CoV-2 infection when combining the two vaccines compared with unvaccinated individuals. And this was the last study, which showed that the combined vaccination was, the mixed vaccination, better than a homologous even the homologous Pfizer combination. And this is a real world observation study. Numbers of 13,000, we have to take that into account. So overall mixing is working. It worked without much data initially when a lot of European countries moved on to it. They really moved very early, while the UK stood by AstraZeneca, and both the immunogenicity safety data is there and the effectiveness data is coming up now that it is effective, the combination is effective. Even in immune deficiency cohort, some data is coming out. So we know that 2 doses are needed. 1 dose is not sufficient. You had, especially with the Delta variant, this was some data produced at that time, UK HPA, and now it has changed its name. So you do need 2 doses. And this was a problem we were having with our immune deficient patients, because a lot of our immune deficient patients may not make a good response to 2 doses. And therefore, third dose came up very early, especially with the immune deficient cohort, the group of patients eyes managed, and some of the cancer patients and the autoimmune patients receiving large doses of immunosuppressive medication. And even with solid organ transplant, you can see that certain people, renal transplant patients, did not get good response after 2 doses. Getting a third dose of vaccine in a certain population group of patients, it increases the response, and at least the antibody response. We have to remember T cell response also is important when taken into consideration. So then I've spoken about mixing and immune deficient. Next, I'll quickly go through some of the booster doses, information about the booster doses that are coming, and what different countries are doing at the moment. Sri Lanka has a booster program in place. So this was the sort of, as Maisha said, I just selected 2 studies, the one with the Zoe COVID study, which was a sort of a pair study, which showed that things seem to be waning as time went by, and about 5 to 6 months was the time that things were changing. And this was the data from that study, but there were other formal studies, and this was the early Pfizer midterm efficacy data. We again showed that there was some waning of responses as time went by. These are the some early studies which made people think about, would we have to give a booster to consolidate the process? So boosters were started very early in Israel. They went into it very sort of very early stage, and you can see from this figure that with the booster dose that was given, they gave vaccination early, first and second dose, and then there was an increase in cases. They went for the booster, and the number of cases started changing after the booster. So it has been shown, this formally study showed the BARD studies, which showed that there was a reduction in admission, severe disease, and death, which was found in this study following the third booster dose. The COV boost study came from Oxford. They have a formula to publish it, but basically the findings is the mRNA vaccine provide a stronger boost effect with low reactogenicity of the vaccine given, irrespective of the vaccine given as a primary course. And that was one of the reasons that Pfizer was taken, or the mRNA vaccine was taken as the third dose or booster dose in the UK, and the process started. The Pfizer or half-dose Moderna was given. It was recommended to give it at 6 months, and initially it was over the age of 50, but now it has been reduced to 40. Everyone over the age of 40 will get a booster dose of the vaccine. There has been some data following that where they found real-world evidence of a significant increase of protection from booster vaccine doses over the age of 50, and this was the UK Health Security Agency study that was that has come out very, very recently. The US has looked at this picture, and they combined different vaccines because they were using Moderna, Pfizer, and the J&J as a additional dose. Reactogenicity was similar across the different vaccines. Booster vaccines increased neutralizing antibodies, and the homologous boost increased it to a lower level compared to heterologous boost. So therefore, the US again has gone over the age of 18 to give booster doses very recently. Previously, they were holding back for some time, but it has gone over the age of